Are you there now?
Yes, sir.
Okay. Now it is or same. Bit. Not much, I
So
think.
is it? Yeah. Last time also it was like that. I think from your end there was some issue.
We wait for a few minutes.
We have 15 participants. Sir, shall we wait or start?
Wait, I think. What do you say?
Yes, so we will wait for one more minute, then we can start, sir.
Deshmukh sir is there, so you can take his opinion initially.
Okay, then I think, sir, we should start.
Yes.
Yeah. So, good evening, everyone. Thank you for joining today's roundtable discussion. And the topic of discussion today is biocontrol, harnessing microorganisms for sustainable future. So we all know that microorganisms are ubiquitously present. So they are present in almost all the different types of environments, and they also we know that they also harbor. They are the common inhabitants of soil. So now, when we talk about biocontrol agents, microbes as biocontrol agents, here first we should it becomes important for us to understand why is there a need to find the alternative. So we all know that today the agricultural sector is expanding because of high food demand, and with this we also have there is an uncontrolled or unregulated use of chemicals, fertilizers, pesticides, fungicides in all types of agricultural fields. So we all know that these are day by day contaminating our environments, our soil, our water bodies. They keep on accumulating because they are not easily degradable. They keep on accumulating in the environment, and they impact all forms of lives. So with this background, here I would like to introduce microbes as one of the most promising uh, sources of biocontrol agents. So, microbes, like there are beneficial microbes that are present in the soil, like we have bacteria, we have fungi, we have viruses that have been found to have some kind of anti microbial activity that is suppressing the plant pathogens which is there in the soil. So, along with the microbes playing other important roles in the soil, like we all know that they play important roles in biogeochemical cyclings to maintain the structure of the soil. So, apart from that, we also know that they are also promoting plant growth, and also we have microbes that are suppressing the plant pathogens that are present in the nearby rhizospheric region of the soil. So, but when we talk about uh, biocontrol agents, microbes as biocontrol agents, along with this, we also have challenges when it comes, however, like translating laboratory success into consistent field performances, since still remains a major challenge. So, that is because of the microbial formulations, the type of microbial formulations. the survivability of the microorganisms in that environment, because there are certain biotic and abiotic factors that contribute to the survivability of these microorganisms, how much these biocontrol agents, these microbes, can naturally or colonize the rhizospheric region, and also limited field validation. So because of these, even though there are breakthrough research in this particular field, but it has been limited to lab or maybe just greenhouse, in greenhouse settings. So when we are talking about field application, this topic still remains challenging and underexplored. So with this, I end my short introduction regarding today's topic. So we have participants who have joined us, and since it is a round table discussion, we expect that participants, they also actively participate in this program. So I would now ask Lami sir to kindly brief about the topic to our participants.
Can you hear me, Upasna, now?
Yes,
Oh,
sir. You're audible, sir.
wonderful.
Clear.
Wonderful. Thank you very much, Upasna, for
Thank you so much,
coordinating
sir.
this roundtable. And good evening, everyone. Biocontrol is very actually important aspect nowadays in the modern science or in the biology, especially using the beneficial microbes when considered as an eco-friendly approach to replace the present chemicals in the form of pesticide or fertilizers or the antibiotics, and this is how actually we are looking for the sustainable development. So what we see is the direct antagonism, in the microbial interaction. The antagonism is one of the important components where one microorganism kills the another one, where we can see actually the Bacillus thuringiensis, the crystal protein, which we are using for killing the Lepidopteris insect, or the Trichoderma, another fungi, which is used for the suppression of some of this weed, right? So there are such a few key mechanisms in the biocontrol, especially in the plant science, I can say. Like one is the direct antagonism, which I was telling. Another is the resource competence, then systematic resistance, nutrient cycling, you know. But nevertheless, as Upasana also pointed out, there are actually the challenges as well as the future outlook, especially in the field consistency, that whatever the data we get in a laboratory may not work in a large scale or in the scale of studies or in the field level, you know. But then definitely a possibility to get more detailed information. Now we can combine these studies with the advanced integration, especially using the tools of genomics, omics sciences, sensor. Now AI ML is another aspect actually is we're waiting for us how best we can actually use. So microbes otherwise we are using, as I told, in the sustainable plant protection, what we call in other words the regenerative agriculture. PGPR, we had actually several roundtable discussion on growth promoting rhizobia recently, and then we actually have given a lot of emphasis on that. Then mycorrhizal fungi also, uh, which I was just now referring to. Endophytes. Endophyte actually has a very good future, which not only helps the plant to stimulate the growth, but even it actually takes care of producing several of the minerals and other phytochemicals. So this actually helps the plant health through nutrient uptake as well as the pest suppression. Bacillus thuringiensis, of course, we are talking quite a lot with reference to controlling the Lepidopteris insect, and very much a proven story which we see especially in cotton, tobacco, and several of these plants where the Lepidopteris insect is the major problem. Otherwise, you will be using the several insecticides, which is not only harm the environment, but then you can actually see the resistance development also. You know, so several such important aspects are there, and as we looking from the window of AMBSI, what are such a frontier areas where now the new budding microbiologist actually can take up. You know, how such, which are the such a useful microorganism that can be actually brought in a practice as a tool, as a biocontrol agent. You know. So this is a very important aspects, for controlling the plants, for controlling this insects, then fungal diseases. We have seen even the few days back, especially with reference to the antibiotic resistance, using the bacteriophages specific, which will target the AMR pathogenic bacteria or virulent bacteria. So several aspects are there. I'm sure that today there are 21 participants. They must be having one or other, what I can say, expertise, or then exposure related to using the microorganisms as a biocontrol agent. So it will be actually very interesting. Even the using the bacteriocins also, which we have discussed as a probiotic organisms, using the bacteriocins, probiotic using against the AMR, right? Several such useful aspects are there which can actually be brought, and not only as a textbook information, or what I can say as a only the theoretical information, but if we can bring it in the practical perspective. Now, if we can develop such a formulations which can be used, not harming the environment, ensuring that our environment also is safe, our livestock also are safe, and even the human health also. So this is what actually in the sustainable development which we are looking for, the better health, better health in all the sphere, the animals, the environment, as well as this human being, right? So it will be definitely interesting how, what are such systems actually we can bring, So Upasana, we can actually go around the people, asking them to share their views and opinion,
Yes, sir.
right? And
Actually,
that will be actually quite interesting actually to know the practical information, in other words. Yes.
yes, sir. Thank you so much, sir.
Over to Upasana now.
Thank you so much,
Yeah.
sir. Now I would like the
Yes.
participants to participate in the discussion. We have 21 participants, so we have Nutan. Nutan is there. Here is there. Divanjali, Sawali. We have Dipti. So Dr. Sarita. Anyone who would like to discuss their research, or who has currently undertaken any kind of master's dissertation or maybe PhD work related to this current today's topic.
Good evening, sir. Myself is Shipra. I'm
Good
working
evening.
as a senior assistant professor in SM College, Bhagalpur, Bihar. Sir, my one of the PhD students is working
Mm.
on this on biocontrol agent on maize plant. So she has grown the maize
Uh-huh.
plant. On that, the
Uh,
effect of few fungus we are observing, like Trichoderma, Aspergillus,
mm.
Penicillium,
Yes.
uh,
Yes.
and we have to find the all biological parameters in comparison to the control variety. How it is is it improving the growth of the plant, or under various biological parameters like length of cob and length of plants, etc. How it is
Exactly.
affecting in comparison to the wild variety. So I just
Very
want,
important topic.
sir,
Yeah, yeah. Please go ahead.
actually I have to go ahead on in this direction. Still, work is going on. So Aspergillus will be like Penicillium and Trichoderma we have targeted first. And how about
Mm-hmm.
Aspergillus? Is it going to help in growth as a biocontrol agent?
Yeah, I think you may have to do some no, control studies,
Yeah,
keeping
yeah.
the different
Yes,
variations, and
two
then
replica we have taken, two
um,
replica of each variety. Like for each
uh,
type of fungus, we have
yes.
targeted 5 different types of fungus, and
Mm-hmm.
pot culture also we are
So you
conducting.
initially perform the yes, in vitro studies you can perform initially, where you will be
Yeah,
able to make out the difference, right?
yes,
Using
sir.
positive and negative control.
Huh.
And once you have such a critical observation from your experimental sample, then you can actually make it scale up, or you can do it in a little larger scale, right?
Yes.
Then I think it is possible that you will be able to. So, but you should have some positive and negative control, of course, without the sample. Positive control, if you can have, then it will be actually interesting to see how best your sample, your experimental material is performing, right?
Mm-hmm.
So that will
Actually,
be actually useful, I guess.
we are conducting
Yeah, please go
parallel
ahead.
field experiment as well as pot experiment.
Yeah, maybe initially what you can do is only the pot experiment, only you can do it in the laboratory scale. Ensure
Yes.
that you see the visible results, because sometimes what happens, we will not be able to actually make it out or verify in a laboratory conditions, because when we go to this field, results are not that reproducible due to one or
Mm-hmm.
other reasons. The what are the changes which we see and all. So due to that reason, better to actually ensure first the laboratory results. Doing in duplicate, replicate, repeating 2, 3 times, and once you are confident, then you can definitely, I think you can then see in the pot, right?
Yes.
And once in the pot, you have confirmed, because all this experiment takes time, right? So
Yes.
multiple experiment has to
Studies.
be planned parallelly, and ensure, yes, and ensure that then your control behaves properly, then your positive is actually showing the difference. And based on that, your 3 replicas, if you can actually prove it, I think you will be able to achieve the your objective. Means your student will be able to do it.
Sir, will you suggest some more fungus
Right?
for
If other,
mycorrhiza also?
maybe Upasna will be able to tell, I think, because
No, no,
he
ma'am.
working on the similar line. Upasna, are you
Yes,
there?
actually, no, actually, we all know that trichoderma is a well-known organism that is used in as biocontrol agent because it is known as a good plant growth promoting. It has a plant
Yes.
growth promoting traits, and also it is used in plant disease suppression. So I will just cite an examples.
Mm.
Ma'am, you can check papers. There are many papers that has been published from this Assam Agricultural University. They have also bioformulations which they have prepared with trichoderma, and they have successful field applications to control plant diseases. So, ma'am, if you want to get the direction in the work, you can check the papers from Assam Agricultural University also. And
Okay.
as you said, you are working in maize, right,
Yes.
ma'am?
Yes.
Ma'am, then have you
Yes.
checked any biocontrol activity of trichoderma against? You can take
It's
you can.
going on actually. It's, we are in process.
Okay, ma'am, then I suggest that you can either you have to isolate from the diseased maize, or else from the literature survey you can see what are the dominant pathogens in this particular maize plant. So
Yeah.
definitely you will find these cultures in MTCC, microbial type culture collection in Chandigarh. So you
Uh-huh.
can order those MTCC cultures, and you can see the impact of trichoderma or what kind of effect it is having against this particular pathogen.
Now, for fungi, I think we have the culture collection center at IRI New Delhi. So approaching there will can be much better. And then you will see now in within a trichoderma you have some 3, 4 different species. So ensuring those species are available, and then they can be against either Fusarium or Rhizoctonia or Pythium, which are actually causing the root rot, or damping off. So
Mhm.
using those pathogen as the indicator, or the sensitive organism will be useful, you know,
Mhm.
so that once you have established those assays in the laboratory condition, then you can actually see in the pot level, and then only the field level to it should be tested. Because the trichoderma not only has the it means insecticidal property, but even they help for promoting plant growth also. So it's such a dual advantage actually trichoderma has, and then you isolating few trichoderma species, characterizing them are actually interesting.
Yes, sir.
Correct.
Yes. We
I
are
think there are
culturing.
well established protocols for. trichoderma. Yeah, you can get one or two culture strain species from other researchers, but you can actually have some collection of the trichoderma because definitely in a different ecological niche we will be able to identify different trichoderma which is not reported before. So that itself can be one interesting study, right?
Mm-hmm.
Yes, sir. Ma'am, you can
Yes,
actually
exactly.
target the local fields if you have, and then isolate trichoderma from there itself. And also, if you want any control study, so
Yes.
comparative study, for comparative study, along with your own isolate, you can check the one that you are actually procuring from any other this culture collection and all. So that kind of comparative study you can do.
Mm-hmm.
And yeah, ma'am, at first you can do the dual culture. These are simple lab experiments that you can do before you move on to pot experiment, even the pot experiment. So there are various parameters that you can check if the organism is actually having any biocontrol activity against any targeted pathogen or not. So for that, ma'am, you can search the literature. You will find ample papers and very simple experiments are there just to kickstart your work in the lab, ma'am. So
Yes,
these dual culture.
culturing on PDA and like identification of the fungus, that is already done, but dual culture like still we are.
If you have got the pure culture, if you have
Yeah.
successfully isolated trichoderma, then if you want to be sure of the organism, then you can go for molecular identification before you start with any work, ma'am.
Okay.
So once you are confirmed with your strain that yes, it is trichoderma, then you can start with your biocontrol activity in the lab.
So from IRA you can procure, right?
Yes, you can.
Yeah, yes, sir. The standard culture, no, fungal culture collection center is there in IRI, but yes, sir, you will see the several trichoderma biofungicides are commercially also available. You know, you have
Yes,
the
sir. It's
in the
commercially
powder form,
available.
liquid formulation. Yeah, so you can even have that also to start with. But meanwhile, you can actually get the trichoderma, no, type culture from the one of this culture collection center. I think in IRI, what we have this fungal culture collection center, I think you will be able to get there. IRI, yes.
So Rhizoctonia also you suggested, right? And mycorrhiza, how about mycorrhiza, sir?
Rhizoctonia is a sensitive organ, sensitive.
Pneumonia is one of the pathogen that you can target. You have then fusarium. Then
Fusarium.
actually, ma'am, first it is better that you first do the literature survey
Okay.
regarding the pathogens present in maize plants, or maybe locally if they are infected. It is also recommended that you isolate those pathogen. That would be better, I feel. And you can meanwhile do comparative studies.
Hmm. Disease may like we have obtained Aspergillus flavus, Aspergillus niger, um, and we have
Yeah, you
uh,
use those culture as a sensitive organism and then you spot the trichoderma. Now, trichoderma, which is actually isolated in IRI, is actually available with MTCC also. In MTCC, they have 5371 culture collection number. So you can actually write to MTCC Chandigarh. They will be
mhm,
able to provide you within a week time because they will have the ready stock. So you need not to struggle much, perhaps
mhm,
for getting the standard culture. And then you can have the commercially available also so that when you do even the field trial, you can always use that one as a positive control and then you can demonstrate how your culture actually is performing. Means your own isolator, your formulations. So that can be actually interesting study, right?
mhm. Thanks, sir.
Uh, Good evening, sir. I'm Jyoti from Pune. Hello.
Yes, yes, you are audible.
Yeah, ma'am. Actually, I'm working on antimicrobial compound. So basically, I got a bacteria from the soil itself. So it is
Uh-huh.
showing the antimicrobial property against the candida. So basically, it is the in a therapeutic
Wonderful.
way. Hello.
Yes, yes, yes.
Yes, sir.
Yes.
Yes, sir.
Very
So
useful.
basically, I'm doing that research in the by taking the main as a candida itself. It is also showing
Yes.
whatever that species I was having, like as aureus and E. coli. It is showing the
Mhm.
antimicrobial property against them also. And after the whole genome sequencing and all, I got to know after that literature reviews and whatever the papers are available, it is having the good agricultural demand, like in the field of the biocontrol and all. So I'm continuing my PhD now. So sir, I need a suggestions, like any top 2 3 microorganisms which are very like demanding in the field of agriculture, which is the serious issues now. So I wanted to check against them, is it is showing any antimicrobial activity against them or not. Can you suggest me some?
Exactly.
Yeah.
What you have to do is you get those indicator cultures or the sensitive organisms or the cultures which are either the plant pathogens or the disease causing organisms, and then you do the plate assays and then demonstrate in the plate that your culture, either the culture filtrate or the cells are actually inhibiting and killing those bacteria, those other sensitive organisms. And once you have performed that, you try to purify that substance to some extent, partial purification, and then see that the activity has increased substantially, you know. And then how we actually can enhance that substance also, because these antimicrobial substances usually will be produced in a relatively lesser quantity, you know. So sometimes they
Yes.
also need the specific media, the growth conditions, some inducer also. Even some of this indicator bacteria itself can enhance, you know. Co-cultivation also is required. You can check
Yes.
those things, and then once you have this one of this actually directly on the plants, you can actually study the co-inoculation. You know, you have one
Yes.
side the sample preparation in the form of either spray, you know, and infected with the plant pathogens, and then see that the severity of the disease actually is reduced, right, compared to the plants where this formulation is not sprayed or inoculated or exposed. You know, if you can show like this kind of experiment at the field level, will be wonderful. Field level in the right. And
Okay.
then one side is characterizing that culture for production of its antimicrobial substance. What is the nature of that substance? Whether it is thermostable, whether it is a peptide, whether it is a organic acid, whether it can be, whether it is producing intracellularly or extracellularly, or in the culture filtrate, or in the solid media only, right? And whether more important is the checking its spectrum, whether it kills
Okay.
beside the gram positive and gram negative, the fungi, and you are telling candida, it is working against candida. So it
Yes.
looks very potent. This is how
Yes, sir.
actually we check the, yes. And when you are trying to purify partially, or purification to homogeneity, if you can actually determine the structure and then elucidate this structure, because we are also working on similar line, look for this ANMR structure or crystallography structure. So it is a long way to go, but then still, because unless and otherwise we determine such a functional group or characterize this such antimicrobial substances, how they usually attack, and more importantly, what is their mechanism, mode of action, whether they are just inhibit the cell wall or the cell division or any important biosynthetic pathways. In fact, in our laboratory, we have for the bacterial system to determine the mode of action of the any antimicrobial substance. You know, we use actually the cell reporter say, and we can actually do a beta galactosidase this one induction with reference to the mode of action of any antimicrobial substance. So if it is working on the cell wall biosynthesis, if it is inhibiting the cell wall biosynthesis, our reporter for the cell wall biosynthesis will give a blue colorations. If it is for fatty acid, it will give a blue colorations. If it is for DNA synthesis, like we have suppose NoHo biosyn, So we use such a positive control or for RNA rifampicin, and then you test your sample, and then you can actually able to correlate the sample which you have antimicrobial substance. What is its mode of action, right? So
Yes.
right now we have only for the bacterial system,
Okay.
but such
Yes.
systems are available for eukaryotes or fungi also, and these
Okay.
are actually very useful with reference to studying the mode of action because you need not to purify the substance. The crude sample also is enough. Then your sample need not to be in a very high amount. Even the nanomolar concentration or micromolar concentration sample,
Yes.
microgram sample also is
Yes,
enough.
sir.
And
Yes.
at a time, you can actually do the several such assays. So you will be able to actually identify what is the mode of action of your antimicrobial substance within a 1 or 2 days. That is what I am trying to tell.
Okay.
Right? Yes. So
Okay.
if you need, you wants to use that system, we will be actually happy to share or extend the technical help for
Yes.
identifying the mode of action of antimicrobial substance.
Yes, sir. Because I'm doing my PhD here. It is a basically not a research lab. We are like
Uh-huh.
BTech. Normally here, maximum BTech students are here, and MTech and
Okay.
PhD. I'm the first PhD come teaching staff here. So we don't
Mm-hmm.
have the much research type of like instruments here. So I'm looking for any collaboration because I went to Bhubaneswar to the SOA. There I have done till the extraction, the
Mm-hmm.
solvent extractions part. Now I am in a phase to purify that compound. So I'm looking
Okay.
for some of the lab who like where that some minimum instrumentation will be there for the extraction, and then I can send for the purification part for the HPLC. After that,
Exactly.
I can go for that structural things and I can send the samples there. So
Exactly.
if anyone like if you know sir, like I can reach to anyone for the collaboration and all. So the
Mm.
mutual benefit will be there. And then please like help me. I will happy to connect. Yeah.
Definitely, definitely. This is what actually this purpose of this round table, so that any researchers who wants to do the advanced work, they get connected with one other laboratory or institute, and then they can actually get benefited through
Yes.
this EMBS platform, right?
Yes, sir.
Where you
Even
are
sir,
doing PhD, by the way?
I'm in Pune, sir, in Ajinkya D.Y. Patil University.
DY Patil.
Ajinkya D.Y. Patil. Yes, sir.
Are you in DY Patil?
Yes, sir. In D.Y. Patil
Okay,
branch
okay.
only.
So
But this
DY
is the
Patil.
branch only. This is Ajinkya sir's like university. Like the younger
Okay, yeah, yeah,
son is there.
nothing.
Yeah. Yeah.
I understood. You can go to NCL or you can go even Symbiosis also. The Symbiosis
Yeah.
University. There also people are working on this line. Or you can
But sir,
directly go to Savitri University also. You can go Savitri Phule Microbiology Department.
uh,
You know,
sir, I
they will
don't have.
definitely.
Actually, here actually, I don't have that much contact. I have done my MSc in 2012, and that time I was in Bhubaneswar. So there I'm having contact, but in Pune I don't have much lab contacts, sir.
Oh, okay,
So,
okay, okay, okay.
so sir, if it is possible, I like in your lab you are allowing, so I can come and work there. I need to take permission from my college, so I can connect you, sir, personally for that. Like if this is the platform where I cannot able to like detail you like how to do and all. If it is
Yeah, yeah,
possible,
yeah.
so I can do. Yeah, sir, can I
Yes,
have
yes, yes.
your number, please? Uh,
I'll put it in the chat box. Okay.
yes, yes, sir, yes, sir. Thank you so much, sir. I'll connect you. Yeah. Thank you,
Yes.
sir. Yeah.
Over
Thank
to
you, sir.
Upasna.
Hello.
Upasna is there?
Yes, yes, I'm there. I'm just connecting my
Yeah,
camera.
I just put my number in the chat box.
Yes, sir.
Yeah, let us go to the next question so that in
Yes,
next 10 minutes we can finish the today's discussion.
right up.
Hello, sir.
Yeah, please.
Am I audible?
Yes, yes.
Sir, myself, Rasika Mhute, and I am from Kolhapur. So, I am a PhD student, and my topic is on studies on plant growth promoting bacteria and their antifungal potential against the phytopathogens. So, now I have completed the isolation of bacteria and their identification.
Mm-hmm.
And in the antifungal activity, I have performed the dual culture assay, as well as I have extracted the lipopeptides from that bacteria. And now I am going to perform the identification of that lipopeptides. So I want suggestion that any other aspect that I can study in this studies. So that will helpful for me.
Very useful. Now, what we were discussing with another participants, almost on the similar line, how you can actually mass
Yes, sir.
produce and then purify that substance and see the spectrum, right?
Okay.
And once you have seen the spectrum, then you can actually look the potency of that substance, the MIC, the MBC, and then you can actually see in some field study where actually in the field
Okay,
or in
sir.
the pot experiments, if you can demonstrate, the activity can be wonderful, right?
Okay. Okay.
And
So
then
I have performed the
you are in Kolhapur University, right?
yes, sir, Shivaji University.
Yeah, yeah. So there's microbiology, even bioinformatics, biochemistry department. They are quite well versed with some of these things. So you can actually discuss with one or other professor there who can actually are
Okay.
working on the similar line, right? So
Okay,
possibility
sir.
is there that you actually progress in this aspect, and a very challenging topic.
Yes, sir.
Yes. How you know it is a lipopeptide?
Sir, actually, the identified bacteria were bacillus species. So most of the bacillus species are lipopeptide producing. So I have made different trials. I have
Hmm.
acid precipitated, and then I have performed the antifungal
Uh,
activity, and they have shown the positive results. So now I have going to do LCMS analysis to confirm the whether they which lipopeptides they are present.
Wonderful. Just ensure that your initial data is clear because you should not misinterpret and it should
Yes,
not
sir.
give you a wrong, a wrong impression because when you are actually taking it
Yes, sir. Sure.
forward, so important
Huh.
that your basic data is actually clear and then quite strong, right?
Yes, sir.
But otherwise, no, looking for this lipopeptide characterization, several studies are there in the literature. You can actually refer those such papers and then see how your work differs. What are the different methodologies they have actually employed? Through that, I think it is possible that you get a good
Okay, sir.
results. What are your latest results, by the way? What are your positive
Uh,
results from your
So I
study?
have found that antifungal activity against the Fusarium oxysporum, then Fusarium graminearum, then Rhizoctonia solani, and
Very good.
Magnaphorta oryzae. So the bacterial strain showed
Yeah, getting any.
inhibitory zone is showing.
Yes, yes. Very good. Just ensure that it is a prominent zone and you
Yes,
check
sir.
with against the different species also. As many as possible species you should check the inhibitory
Okay, sir.
zone so that you get actually overall the spectrum of your substance and then see that substance can be actually produced in a large scale, right?
Okay, sir.
So beside doing the conventional agar well diffusion assay, you can go for the liquid broth, check the minimum inhibitory concentration, then see
Okay.
the mechanism of action, whether
Okay.
intracellular solute release is there or whether in cell wall damage is there
Okay.
or you see yeah, growth only what we see in the bacteriostatic or bactericidal, right? So you can actually
Right.
ensure those kind of observations so that you
Okay, sir.
have the better insight from your studies. Uh, there are several such publications and then information available, but since your culture is your new, so I think it is possible to looking the new findings, I would say, right?
Okay, sir. Thank you, sir.
Yes,
Yes.
sir. And I think more that you can try on bacterial pathogens as well. I think targeting bacterial pathogens. Are you not using any bacterial pathogens?
No, my study topic is only focused on antifungal pathogens.
Okay,
Sorry,
so it's
fungal
just
pathogens.
antifungal.
Exactly. Very promising results.
Yes, sir.
Now, this is which year of your PhD, by the way?
This is the third year, sir.
Yeah, so you
I
should
have
be
actually
actually able
completed
to
the.
pinpoint to.
Sorry, sir.
Uh-huh. No, you should be able to characterize your substance, the nature of the substance, and how prominent it is, like this way. Prominent means
Okay.
potent. How the substance
Yes.
is potent. So you should actually partially characterize also.
Yes, sir.
Now there are several such studies associated with this kind of things. Definitely, I
Yes,
think you will be able to contribute in a
sir.
very
Yes,
good
sir.
information.
Sure, sir.
Very good. Next, Upasana, are you there?
Yes, I am there.
Wonderful. Go to the next participant.
Let's see. So I will just share the feedback form with them.
Yes, definitely.
Oh, this has not been shared today. The feedback link. So today's discussion was very engaging actually. So I hope our participants have benefited from this discussion.
Hope so.
Yes, sir, and got some direction in their research work.
Exactly.
So any other participant would like to?
Yeah, they can share their experience, opinion. You can just name one by one slowly.
Yes, we have Shivam
Yes,
Kumar.
ma'am. Yes, ma'am.
Yeah, you are pursuing what degree right now?
Basically, ma'am, I am Shivam Kumar from Kalinga University as second year BSc Biotechnology student. In last few in May or June month, I pursuing my internship. During that time, my HOD suggest me or my guide may suggest me you have to start your research work. Uh, from this time then and last year you have to submit a research paper and the dissertation. That's why I joined this session because I thought the session was very wonderful and I got many ideas from the many researchers and many professors. Thank you
Yeah,
so
I
much.
hope the discussion today proved helpful for you. I hope you will
Yes, ma'am. Very, very helpful for
benefit
us,
from
ma'am.
this. You
But,
will
ma'am,
benefit
I
from
got
this.
many ideas how people do their researches. Many I get idea like how to approach any research work. Basically, ma'am,
Yes,
I
yes,
choose
that is
the
really
topic.
important to start. Yes.
Basically, ma'am, I choose the topic like phytochemical analysis of Ipuma equitia. On during my internship,
Okay.
I performed my phytochemical analysis. Then I now am targeting to write my research work like paper. Thank you, ma'am.
Okay.
Mm.
So you stay connected. You can join every Tuesday. We have this roundtable discussion, so you can join
Yes, ma'am.
to get more into various
Yes, ma'am.
topics.
I got many ideas. Like some of my ma'am told that their students are doing research on maize. I got ideas how to approach a research work or any laboratory work. Basically, ma'am, I am
Which
yes,
is
ma'am.
very
Basically, I am very junior than all you guys. Thank you so much for this session, ma'am.
Mhm. Thank you for joining. Yes.
Thank you, ma'am.
Any other participant would like to interact?
Dr. Premila is also there. She also worked on the antimicrobial substance from bacillus. She can tell you her
Okay.
opinion.
Dr.
Good
Pramila.
evening, everyone. I am also very glad to attend this session. I worked under the guidance of Dr. Prakash sir, and I have completed my PhD and I worked as a senior project associate under his guidance. It's very awakening that all the uh, graduates and postgraduates and the fresh PhD holders are getting the opportunity to attend this kind of session, and sir has given a very brief and clear clarity about how we can pursue our research when we are stuck with our results by comparing with the positive control, and how we can take it forward, how we can compare the literature study, and how we can take the inputs. Everything sir has mentioned for the beginners. So I hope this session is very helpful to each one of us, and I'm very thankful to sir also that he has taken his precious time to spend with all the students and the upcoming students. Thank you, sir.
Thank you so much.
Okay. Not as an acknowledgement, but then you have to tell your experience, opinion about no this roundtable. That is what actually we wanted. You know
Yeah,
how
this
we can actually
roundtable is
improve or this can be useful.
Yeah, yes, sir. This session is also important to get some basic idea about research and what things can be shared among ourselves, because everyone is not aware of everything. So this roundtable makes us to learn new things and how we can improve our knowledge also. So this is a very good session, sir.
Hmm. Going to other participants. Yeah, Upasna, you can conclude. I think
Yes, sir.
Sir,
somebody is telling.
Yes, somebody
Yeah,
is there.
yeah,
sir,
he
me
has
only,
some.
Jyoti only.
Yes, please continue.
Sir, I just want to ask a simple thing, which nowadays many students are doing. Even sometimes I also do. Like whenever we are trying to choose a target microorganisms, like you said that
Mm,
you check the control microorganisms, like whatever that against we will check, we are using the chat GPT. Very frankly, everyone is using the AI, and then they are checking that which are in the trend and all. And after that,
mm,
whatever the literature review I am doing, according
yes,
to that, whatever our normally research rules are there. But I
mm,
don't think that it is the too much correct way, because whatever that internet will give, it will give in the publications point of view or any point of view. So what is the correct way to choose the target microorganisms, especially in the a case of plant related, because therapeutics related we can go for directly the case studies, what are the numbers of the that things are there. But regarding the plant things we are not getting, and I'm quite confusing, like shall I go for the any plant specific, like anyone is doing in the maize, anyone is doing in that rice or strawberry or something, or we should choose one criteria where it will fit or it will be nice. So this will be the one issue. Like randomly we are doing to just complete the PhD or complete the things. But what is the market things? So once it will come to the market, there is the gap. So how to get that gap, sir? Like how to find that question and
Upasna, I think, is now role model for guiding you people because she is a fresh PhD. So she will be able to precisely explain you.
Thank you, sir. Like, I just wanted to know from where have you isolated the organism?
I have like I got this organisms from the main rhizosphere.
What plant? Which plant you mentioned? Just
Mint, pudina.
okay. No, I feel like when you are targeting any organism, the antimicrobial property of your isolate against any organism, just doing it randomly would not be a very wise approach. What you can do is you can see your local area if they are
No,
cultivating any major crops, or if it is suffering from any kind of particular disease, then the causal organism of that disease you can target likewise, I feel, from my opinion.
no, not from the isolation point of view, ma'am. Like I'm not asking about the screening or sampling. Right now, I'm having one compound. I wanted to
Yes.
test against the
Yes, the
food or all that. So for that area randomly we should choose or any specific reasons we should have to get that, because there are so
you have
many things are there. So randomly we can choose anyone, but exactly how to get that data? Therapeutics way we are getting like disease numbers and all, but in the case of plant. I'm not getting it is somewhere like mentioned that these many plants numbers are like having this disease or something as a like,
you want the plant disease information, you mean to say?
yeah, like how to choose that target plant or microorganism. For example, someone says that I'm working in the maize, someone is working in the rice. Okay, even is strawberry, we can do that disease and then we can cure. So how to choose that which one we should go? Because
Yeah,
like
that's
in
what I
disease,
feel
I'm
like.
getting.
Yeah, like for me, see, I'm just giving an example from my end. I am in Assam. So in Assam, if you see, tea production is one of the major uh, crop that they are growing. So tea is the major economy from where it comes is from tea in Assam.
Okay,
So
okay.
here we are majorly focusing on tea plant pathogens.
Okay. Okay. So
So
zonal wise, we can go.
yeah, so some important agricultural crops in your local area or your region, so that way you can have more impact. Or if there is any new pathogen that has been reported, so you can also test your antimicrobial activity of your particular compound against that pathogen. So that way the approach would be more good, I feel, just that not randomly targeting any pathogen. So if suppose
Yes.
you are targeting suppose let me say suppose tea only, if you are targeting any fungal
I
pathogen of the
got,
tea.
ma'am. Yes, ma'am. I
Yeah.
got the clue you have said just now that you can go for the recently added pathogen. So I
Yes.
got the clue. I'll do that. Thank you so much, ma'am. Yeah.
Yes, yes. Yeah. Okay. Thank you.
Yeah. Thank you.
Hello, sir.
Yeah, we can conclude now. You just
Okay.
see in the chat box, there are some 10 people have commented. You can quickly go through that and then we can conclude.
Yes, actually, they are inquiring about the feedback form, sir. So this time,
Yes.
yes, I have not actually received the link because generally I receive the link in the group. So today I am not being able to share the link actually, sir. That is the problem.
So how do we means record those things?
So these are
Or how we can actually.
these are already like recorded only, sir. This is like already recorded. These discussions are already recorded, but the feedback link, sir, I have not received today from the chairperson itself, so I'm not able to share with the participants, sir.
Okay. Well, during next roundtable, we will make sure that they have been given the,
Yes, yes.
the certificate. Now you have given the link for the certificate. That is combined, right?
Yes, sir.
So
From
they
the
will not be able
feedback
to get
link
the
only,
certificate now.
sir, for today's, yes, sir.
Okay,
Because
okay. Yes. So we will somehow make sure that their participation is considered, and then we will give the certificate next time.
Yes,
Right?
definitely, sir. Yes, sir.
Yes.
I will talk to
You
the
can just go through the
regarding the
chat
same.
box. Yeah, exactly.
Yes.
You can go through the chat box and then you can conclude.
Yes, sir. And then apart from that, sir, there are not any important messages regarding.
Wonderful.
Yeah. Okay, sir. Then thank you all the participants for your participation in this important discussion regarding an important topic, which is biocontrol. And there are much more that needs to be discussed. I feel, sir, right? Sir, like we have just discussed
Definitely.
aspects. Yes, sir. We have so many other different aspects that can be discussed under this
Yes.
topic. So we have just today just biocontrol agents. So we did not even touch the entomopathogenic fungi, which is very important in controlling pest. So these aspects, because of time constraint, yes, sir, we could not discuss only, but I hope this will continue again in the next roundtable discussion on Tuesday. So thank you all the participants, and special thanks to our sir, Prakash Shalami, sir. Thank you so much, sir, for your time and your valuable suggestions
Thank you.
to the participants. And
Thank you.
okay, sir, we'll meet. Sir,
Thank you.
yes, sir, I will connect to you through the WhatsApp, sir. I have one work which I wanted to discuss. So tomorrow or day after, yes, sir, I will discuss. Okay, then. Thank you,
Definitely.
sir. Yes.
Thank you. Thank you, everyone, and bye.
Thank you, sir.
See you next time.
Thank you,
Next
sir.
time.